Myself, BABU, Botany faculty. Today I am going to explain about topic of Bryophytes. So bryo means moss, phyta means plants. Which plants growing as a dense patches like a condition. That means grouping the plants very close to each other. So these plants is known as a Bryophytes. This growing a dense patches like a condition on a moist soil, then tree trunks, then rocks, etc. This plants is known as a bryophytic plants. These bryophytes compared to algal members highly evolved condition. These bryophytes occupy between algae and pteridophytes. So bryophytic plants are occupied between the class of algae and pteridophytes. Bryophytes are first land inhabiting habitats of plants. That means first terrestrial plants are bryophytic plants. Study about bryophytes is known as a bryology. Study about bryophytic plants is known as a bryology. The term bryophytes coined by Robert Brown scientist. The term bryophytes coined by Robert Brown scientist. Father of bryology is known as a Hedwig scientist. Hedwig scientist is known as a father of bryology. These bryophytic plants are non-vascular cryptogamic plants. Non-vascular cryptogams. Bryophytic plants are non-vesicular cryptogams. That means vesicular tissues, xylem and phloem conducting tissues are absent. These are non-flowering plants. So no flowers and no seeds are present. This is also known as a atrachophytic plants without vesicular tissues, xylem and phloem. These bryophytic plants are first archegoniate plants. So bryophytic plants are first archegoniate embryophytic plants. Archegonia first developed in bryophytic plants. In algal members, female sex organ is known as a oogonium. From bryophytes, pteridophytes in gymnosperms, female sex organ is known as a archegonium. That's why these are the first archegoniate plants. Because of this embryo condition also present in bryophytic plants. In algal members, embryo stage is absent. Zygote act as a meiocyte. Here embryo stage is present. That's why first archegoniate embryophytic plants are bryophytic plants. Then generally bryophytic plants growing on moist and shady localities. That means moist soil, then moist or wet condition of rocks, then tree trunks, etc., then burnt soil, etc., etc. Some bryophytic plants may be aquatic in condition also. Then in bryophytes, the plant body is a thalloid condition, or leafy axis is present. In bryophytic plant body is a thalloid or leafy axis is present. Thalloid means not differentiated into true root, stem and leaves. Then in some bryophytic plants leafy axis is present. In thalloid condition mainly liverworts. Then in leafy axis mainly moss plants, that means Bryopsida plants. Both the thalloid condition and leafy axis is present. In leafy axis maybe leaves are present. These leaves are very small leaves. and sessile condition. That means without petiole. These leaves are directly attached to stem like or branches like structures. Then these leaves are ovate condition. Then cute apex is present. Like this one. So these leaves are sessile condition, then ovate condition, then cute apex is present. Leafy axis mainly present in only mosses in Bryopsida plants. Then in a bryophytic plant body is a haploid condition. That means N stage is present. It is a dominant stage in bryophytic plants. It is a photosynthetic nature. That means prepares the food materials. Then in bryophytes, reproduction by mainly two methods. It is vegetative reproduction, then sexual reproduction. Bryophytic plants reproduce mainly by vegetative method and sexual method. So vegetative reproduction by fragmentation, then formation of adventitious branches or adventitious buds. Adventitious branches are adventitious buds. Then bulbs, then tubers, then gem. So vegetative reproduction by the fragmentation method, by the formation of adventitious branches, adventitious buds, bulbs, tubers and gemmas. Then sexual reproduction. In bryophytic plants, sexual reproduction mainly oogamous type. In algal members, isogamy, anisogamy, then oogamous present. From the bryophytes in all plants, oogamous type of sexual reproduction is present. Then clear cut sex organs are here present. Then in algal members, sex organs are unicellular, non-jacketed condition, but in bryophytic plants here multi cellular condition, then jacketed stalked condition, jacketed and stacked. In bryophytic plants, sex organs, both male and female sex organs are multicellular condition, jacketed, covered by a single covering coat. These are the sterile cells. Then stalked condition, by the stalk, these are attached to male thallus or female thallus, both are present on the same thallus. So multicellular, jacketed and stalked condition. In algal members, sex organs are unicellular, non-jacketed in a condition. Then in bryophytic plants, male sex organ is known as a antheridium. Then female sex organ is known as a archegonium. Antheridium and archegonium. So male sex organ is called antheridium, female sex organ is known as a archegonium. Then antheridium generally club shaped condition. So antheridium generally club shaped condition. So anther wall, operculum and stalk. Single layer of jacket is present. So within this jacket maybe sperma cells are present. That means antherozoids. Number of antherozoids producing within this antheridium. These are biflagellated condition, spirally coiled condition, biflagellated, then spirally coiled condition. Anthrozoids or sperms are biflagellated, spirally coiled condition. Then after mature of these sperms, then release into substratum by the help of water. This may be reached to archegonium. Then fertilization takes place. Then archegonium generally flask-shaped condition. Archegonium generally flask-shaped condition. It is differentiated into three parts: stalk, venter, and neck. Neck generally single-layered condition. Then 5 to 6 rows or 5 to 8 rows of neck canal cells are present. Then the middle portion is a venter. It is a 2 or 3 layered condition. All of the sterile cells. Within this venter, 2 or 3 venter canal cells are present. These are the venter canal cells. These also sterile in a condition. Then only one egg cell is present. Egg cell only fertile in a condition. Archegonium is a flask-shaped condition. It is differentiated into three parts: stalk, venter, and neck. In neck, rows of neck canal cells, 5 to 6 rows or 5 to 8 rows of neck canal cells are present. These are the sterile cells. In middle portion, venter is present. It is a 1 to 2 layer condition. Within venter canal cells, 2 or 3 is present. This also sterile condition. Only one egg cell is present. It is a fertile condition. By the help of stalk, these are attached to female thallus, or both are male and female sex organs are formation on the same thallus. So these sex organs are stalked condition, zygoted condition, and multicellular condition. When these anthrozoids sperms are mature, these are liberated, released from this antheridium into substratum by the help of water. These are the flagellated motile condition. Swim in water, reach the neck of archegonium. While this time, these neck canal cells, ventral canal cells may be disintegrated condition. That means degenerated, may be release some chemicals. By the help of chemicals, K plus and proteins. K plus and proteins. These K plus and proteins attract these anthrozoids. Anthrozoids attracted by K plus and proteins then enters into neck. This type of sperm movement is known as a chemotactic movements. Chemotactic. So in bryophytic plants, sperms movement motility by the chemotactic. That means sperms enters into neck or archegonium by the attraction of chemicals K plus and proteins. This movement is known as a chemotactic movement. Then these anthrozoids travels through the neck, then reach to venter, then reach to egg cell. Then fertilization takes place. This fertilization is known as a syngamy. Fertilization is known as syngamy. Syngamy is nothing but a fusion of male and female gametes. By the results of syngamy, here form the zygote. So zygote is a diploid condition. Both the anthrozoids and egg cell is a haploid condition. By the fusion of this haploid egg and anthrozoids form the diploid zygote. So it is always a 2n condition. In bryophytic plants, this zygote immediately undergoes to mitotic division, then form the embryo. Here no resting stage. That means a zygospore condition not present in bryophytic plants. So results of syngamy from the zygote. Then zygote covered by a thick wall coat. This wall coat is known as a calyptra. Wall coat is known as a calyptra. Sterile cells are present. So zygote covered by wall layer. This wall layer is known as a calyptra. Calyptras are the sterile cells. Then zygote immediately undergoes to mitotic division. There is no resting stage. Not present a zygospore stage. Then develops into embryo by the results of mitotic division. Then embryo develops into sporophyte. Embryo develops into sporophyte. Sporophyte also 2N in a condition. Then sporophyte are differentiated into three parts. These are foot, seta and capsule. Foot, seta and capsule. Sporophyte differentiated into foot, seta and capsule. So in capsule, spore mother cells are present. In capsule, spore mother cells are present. These spore mother cells undergoes to meiotic division, then produce the haploid spores. Produce the haploid spores. Then by the germination of these haploid spores produce the new thallus of bryophytic plants. These type of fertilization oogamy is known as a zooidogamous oogamy. Zooidogamous oogamy. In bryophytic plants, type of oogamy is a zooidogamous oogamy. That means the motility of the sperm by the help of water, then by the attraction of chemicals, chemotactic movement reach to archegonium, enters into archegonium, then fertilization takes place. This type of oogamy is known as a zooidogamous oogamy. So heteromorphic, these are morphologically distinct in a condition. Both a sporophyte, gametophyte with a distinct morphological stages are dissimilar in a condition. But a gametophytic stage is a dominant condition. Sporophyte depends or present on gametophyte partially or completely, depending on sporophyte for a nutrition. It is a semi-parasitic condition or completely parasitic in a condition. Life cycle in bryophytes are haplodiploidic life cycle is present. Haploid and diploid stage in alternate manner. Haplodiploidic life cycle is present. Then bryophytic plants generally known as amphibians. Bryophytic plants is known as amphibians in the plant kingdom. Why? Because bryophytic plants are first land plants, first terrestrial plants. At the same time requires water for sexual reproduction. That means these are present on land. At the same time requires the water for sexual reproduction. That's why this is known as amphibians in the plant kingdom. These bryophytic plants are adapted to both the land conditions and water habitats of conditions. So what are the adaptations to land habitats? What are the adaptations to land habitats? These root-like structures, rhizoids are present in bryophytic plants. Except mosses, true root stems and leaves are absent. Root-like structures, stem-like structures, then leaf-like structures are present. Root-like structures is known as rhizoids. These are unicellular or multicellular in a condition. These rhizoids are responsible for absorption of water. True roots are absent. It is a one condition adaptation to land. Next one, zygote formation within archegonia. Zygote formation takes place within archegonium. Then zygote to embryo. Zygote to embryo. Zygote formation by the fusion of male and female gametes. These male and female gametes are jocketed condition. In algal members, non-jacketed condition. In bryophytes, jacketed condition. These are four characters may be adaptation to land habitats. So bryophytic plants, amphibians are adapted to both land habitats and water habitats. Adaptation to land habitats are rhizoids. True roots are absent for absorption of water. Then jacketed sex organs, both male and female. Then zygote formation, zygote to embryo formation. These characters are adaptation to land. Then adaptation to water habitats. Adaptation to water habitats. Here also true roots are absent. True roots are absent. Instead of true roots, rhizoids are present. So conductive tissues are absent. That means vascular tissues are absent. Xylem and phloem, conductive tissues. Why? Because these are mostly growing in moist condition, maybe in aquatic condition. Body structure also organization very simple in a condition. So that's why there is no clear cut mechanical tissues and conductive tissues are absent. Next one. So chemotactic spore movement. Chemotactic spore movement. These are three characteristic features are adaptation to water habits. Next one, smallest bryophytes is juices. So approximately 5 mm condition. Then largest bryophyte is a Dawsonia. Largest bryophyte is a Dawsonia. Bryophytic plants are very small, simple structure, approximately 2 to 10 centimeters. So smallest bryophyte is a Jups is 5 mm. Then Dawsonia maximum 10 or 15 centimeters. Maybe belongs to Bryopsida mosses plants. Then based on the characteristic features, these bryophytes are classified into, division of bryophytes are classified into three classes. These first one, aquatic oxida. Then second one is a Anthoceratoxida. Then third one is a Bryopsida. Based on characteristic feature, bryophytes are classified into three classes: hepatic oxida, Anthoceratoxida, Bryopsida. Hepatic oxida is known as a liverworts. Then Anthoceratoxida is known as a hornworts. Then Bryopsida is known as a mosses. Liverworts, hornworts and mosses. Why it is known as a liverworts? The thallus appears like a liver-like condition. So liver, what is nothing but a small plant. What means smaller plant? The thallus body, the plant body of bryophytes in hepatic oxida appears liver-like condition. That's why it is known as a liverworts. Then sporophyte in Anthoceratoxida appears like a horn-like condition. That's why this is known as a hornworts. In Bryopsida, plants growing a dense patches like a condition. That's why this is known as a mosses. Then examples of hepatic oxida, Riccia. Then Markantia. Then Pelia. Then Corella. So Riccia, Markantia, Pelia and Corella. Then hornworts example Anthoceras. Then Bryopsida examples Filaria, Polytrichum, Sphagnum. Filaria, Polytrichum and Sphagnum. So we have to study about all these characteristic features one by one. So first one coming to liverworts. So work means smaller plant. So liver means the thallus appears, the plant body appears liver-like condition. That's why this known as a liverworts. So characteristic features. Liverworts generally growing in moist and shady localities. That means bank of streams. Bank of streams. Then bark of trees, then marshy ground, then deep in wood. So these are generally growing in shady and moist habitats, bank of streams, bark of trees, marshy ground, and deep inside the wood. Then body organization is generally thalloid condition. In liverworts, body organization is thalloid. That means not differentiated into true root, stem, and leaves. Best example, Markantia. We are going to study about the characteristic features of a Markantia. In Markantia, plant body is a thalloid in a condition. So plant body of a Markantia. So it is a thalloid condition, not differentiated into true root, stem, and leaves. Maybe root-like structures, rhizoids, then stem-like structure, then leaf-like structures are present. It shows the dorsiventral flat-ended structure, maybe prostrate condition on a substratum. Dorsiventral. So ventral side and dorsal side, both the sides are present like lower and upper in a condition. It shows the dichotomously branched condition. Dichotomously branched condition. This is a total thallus very close to the substratum. That means appressed to the substratum. So in Markantia, plant body is a thalloid condition. It is a dorsiventral flat-ended structure, dichotomously branched condition. In this thalloid on stem-like structure, leaf-like appendages are present. So it is a leaf-like appendages. These appendages has smaller leaf-like structures with two rows of arrangement. So leaf-like structures arranged in two rows. Each row has a one apical notch. Each row has a one apical notch, then one midrib. So parenchymatous body is present. Both male thallus and female thallus is a separated condition. Sex organs also formation separately. Male thallus, male sex organ, and female thallus, female sex organ. This condition is known as a dioecious condition. This condition is known as a dioecious. Dioecious is nothing but male and female sex organs formation on a different thallus, male thallus and female thallus in a condition. Then reproduction by vegetative reproduction, then sexual reproduction. Vegetative and sexual reproduction. So vegetative reproduction. In Markantia, vegetative reproduction mainly by the fragmentation method. That means in adverse condition, by or due to the by effects of some adverse condition, the plant body may be split into smaller pieces. Each piece have the ability to develop into a adult Markantia thallus body. This is known as a fragmentation or by the formation of gemmas, or by the formation of gemmas. These gemmas are multicellular condition. These gemmas are multicellular. Their green condition, their biscuit shaped, multicellular, green and biscuit shaped asexual buds. These gemmas are formation on receptacle of a thallus body in a cup-like structures. This known as a gemma cups. Gemma cups. After complete development of these gemmas, so release from the thallus into substratum when conditions are favorable by the germination are developed into a adult Markantia thallus body. So these are gemmas. So generally gemmas are egg-shaped condition. In middle portion one notch is present. One gemma then produce the two thallus, two daughter Markantia plants. So gemma generally egg-shaped condition between or middle in portion one notch is present. So this one can capability to produce the new daughter Markantia plants. This one can have the capability to produce the new thallus of Markantia. Each gemma will produce the two daughter plants. Next one, sexual reproduction. Sexual reproduction by the oogamy type. Sexual reproduction by the oogamy type. So both male and female sex organ. So male sex organ is known as a antheridium. Then female sex organ is known as a archegonium. In Markantia, both the male sex organ and female sex organ formation are present on the different thallus. That means a male thallus and female thallus. Male thallus and female thallus. So male sex organ on formation of male thallus, female sex organ formation of female thallus. So these sex organs are stocked condition with a receptacle. Both male and female are stocked and receptacle. These sex organs are formation on a leafy-like structure. Leafy-like thallus structure. Stocked receptacle. This stocked receptacle of sex organs is known as a gametophore. Gametophore. So stocked receptacle of sex organ is known as a gametophore. So gametophore with receptacle on female thallus is known as a archegoniophore. Archegoniophore. That means a gametophore with a female thallus. This is known as a archegoniophore. Then receptacle, stocked receptacle with a male thallus. This is known as a antheridiophore. Archegoniophore and antheridiophore. So these archegoniophore, these receptacle as a 9 cylindrical rays. 9 cylindrical rays with archegonia. So receptacle of female sex organ has a 9 cylindrical rays with archegonia. Then the receptacle of antheridiophore, so 8 lobed condition. 8 lobed condition. Each lobe with the alternate of A chamber and antheridial chambers. Receptacle of so antheridiophore, the receptacle is a 8 lobed condition. Each lobe with the alternate of A chamber and antheridial chambers. So each antheridial chamber contains one antheridium, club shaped antheridium. This antheridium produce the more number of biflagellated sperms or antherozoids. Then rhizoids are present on a ventral surface. Rhizoids are present on ventral surface. These are unicellular in a condition. So after mature these antherozoids then release into substratum, then by the help of water, swimming in water, then by the attraction of chemicals reach to archegonium, then travels through the neck, then reach to venter, then fertilization takes place, then form the zygote. Then zygote develops into embryo, then embryo develops into sporophyte. Embryo develops into sporophyte, then sporophyte differentiated into foot, then seta, then capsule. Foot, seta and capsule. In capsule, spore mother cells are present. These spore mother cells diploid condition undergoes to meiotic division, then produce the haploid spores. Then this haploid spore directly germinate, then form the new Markantia plant body. This type of oogamy is known as a zooidogamous oogamy. In this life cycle, heteromorphic alternations generations, both the gametophytic and sporophytic generations with distinct morphological phases, haplodiploidic life cycle. Then Anthoceratopsida. So in Anthoceratopsida, the characters are maximum similar to Hepaticopsida, but in Hepaticopsida, the spores released by the special structures, that means elaters. In Anthoceratopsida, pseudo elaters are present. Rest of the all characters may be Anthoceratopsida similar to Hepaticopsida. Next one, Bryopsida. So, characteristic features of Bryopsida, that means bryophytic plants. In liverworts, spore directly germinate to form the new thalloid body, but in mosses plants, spores not directly germinate, indirectly germinate to form a vegetative thallus. This vegetative thallus is known as a protonema. This vegetative thallus is known as a protonema, may be photosynthetic condition without sex organs. In Bryopsida, in mosses plant, spore indirectly germinate to form a vegetative body of structure. This is called a protonema. This protonema is a filamentous condition. This protonema is a filamentous, then branched prostrate condition. It is a filamentous branched prostrate condition. Then this protonema will form the adventitious buds. On branches of this protonema, then form the adventitious buds. These buds then gives rise to new thallus or form the secondary protonema. Secondary protonema. This secondary protonema with the leafy axis is present. So secondary protonema with a leafy axis. In this condition, maybe sex organs are present. So male branch and a female branch. On male branch, male receptacle, male sex organ. On female branch, female receptacle, female sex organs are present. Both these sex organs are present on the tip of the branches. Tip of branches. Next one. It is a sporophyte. This sporophyte differentiated into foot, seta, and a capsule. Sporophyte differentiated into photoseta capsule. It is a gametophytic plant body. So N condition, it is a sporophytic plant body. 2N in a condition. Then vegetative reproduction in a Bryopsida plants by fragmentation. Then secondary protonema, bulbs, their germe formation. Vegetative reproduction in Bryopsida plants by the fragmentation, by the secondary protonema, bulbs and gemmas. Then sexual reproduction by oogamous type of distinct male and female sex organ, antheridium and archegonium. So antheridium generally club shaped condition, archegonium is a flask shaped condition. Then antheridium produce the number of biflagellated sperms or antherozoids. Then archegonium with one egg cell with the stalk, venter and neck is present. When antherozoids are mature condition, these are liberated from this antheridium, then reach to mouth of archegonium, that means chemotatic attraction, then fertilization takes place, then form the zygote, then zygote to embryo, then embryo to sporophyte. So 2N in a condition. Sporophyte in Bryopsida highly evolved, more elaborate than the liverworts plants. Here also sporophyte differentiated into foot, seta and a capsule. Foot, seta and a capsule. Sporophyte differentiated into foot, seta and a capsule. Then capsule again differentiated into Apopasis, theca and a operculum. So capsule. So in Bryopsida plants, capsule differentiated into three parts. So first one is a Apophysis. Apophysis, then theca and operculum. Capsule in a Bryopsida plants differentiated into Apopasis, theca and operculum. Apopasis is a base pod. It is maybe green in color, can perform the photosynthesis, prepare the food material. But for photosynthesis, it requires water and mineral nutrients. That's why it is a semi-parasitic condition. Sporophyte in Bryopsida plants, semi-parasitic on a gametophytic generation, not a complete parasitic condition. Then theca is a middle portion. It is a fertile region in capsule. Fertile region in capsule is a theca. It is also differentiated into different parts. Single layer epidermis is present. Then below this epidermis, two layers hypodermis is present. In central portion, sterile cells are present. This is known as a columella. So columella is a sterile in a condition. Then both sides of this columella, spore sac is present. Within this spore sac, spore mother cells are present. So all parts in this capsule are diploid condition. So spore mother cells also diploid in condition. Then this spore mother cell undergoes to meiotic division, then produce the haploid spores. Then the tip apical part is known as a operculum. This operculum connect with this theca by the sterile cells. This is known as a annulus. It is a cap-like structure. Within this operculum, specialized teeth-like structures are present. These known as a peristomal teeth. So total 32 peristomal teeth are present. These are arranged in two rows. In each row, 16 peristomal teeth are present. Outer row, then inner row. So outer row is known as a exostome. Inner row is known as a endostome. Exostome and endostome. This exostome is a hygroscopic in a condition. Then inner peristomal teeth is a non-hygroscopic in a nature. When these spores are mature, then this annulus is shrinks. When annulus shrinks, this outer peristomal teeth bend down outwards in condition, then fall down this operculum, then release the spores into substratum. Then by the germination of spores develops into a new bryophytic plant body, that means Bryopsida, especially in mosses, Funaria. These all characteristic features belongs to Funaria plants. So in Bryopsida, in mosses, the spore indirectly germinate to form a vegetative stage of protonema, then protonema to secondary protonema. On secondary protonema, leafy axis is present. So these leaves are arranged spiral in manner. These stages contain the sex organs, both male and female sex organs on same thallus. So it is a monoecious in a condition. Morgans is a dioecious. It is a monoecious in a condition. Then economic importance of bryophytes. Economic importance of bryophytes. So bryophytes has a less economic importance. So this mainly play a important role in plant succession. Important role in plant succession. Along with the lichens, this play important role in plant succession. These may be decompose the rocks, make the soil for growth or succession of higher plants. So that's why these Bryopsida plants have their high ecological importance, play important role in plant succession. Then these bryophytic plants growing as a dense patches like a condition on soil, mat like a condition, dense mat like a condition. That's why these are prevents the soil erosion. These are prevents the soil erosion. So bryophytic plants prevents the soil erosion. Next one, number of bryophytic plants useful as a food for birds, some mammals, etc. Food for birds and some mammals. Next one, in bryophytes, sphagnum. Sphagnum is known as a peat mass. Sphagnum is known as a peat mass. This can useful as a fuel. This we can use as a fuel. Then this peat mass useful to production or obtain the some chemicals. This ammonia, then ethyl alcohol, then paraffin, etc. Ammonia, ethyl alcohol and paraffin. This peat moss generally have the capability to water holding capacity. Due to retain water holding capacity, we can use this peat moss for transportation of living material. We can use this as a for transportation of living materials. Then life cycle of these bryophytic plants. Haplodiploidic life cycle. Life cycle of bryophytic plants is a haplodiploidic. Bryophytic plant shows the heteromorphic alternations of generation. Both gametophytic and sporophytic generations morphologically distinct condition. This is known as a heteromorphic alternations of generation. Bryophytic plant body is a haploid condition. It is a dominant phase. Bryophytes. Haploid condition. This form the male and female sex organs. Male and female sex organs. N condition. So male sex organ is known as a antheridium. Female sex organ is known as a archegonium. Antheridium and archegonium. So both are haploid in a condition. Then antheridium produce the sperms are antherozoids. Antherozoids. Haploid condition is produce the egg cell. So maybe by the mitotic division. So male sperms or antherozoids are egg. Then fertilization. Syngamy takes place. Results of this syngamy form the zygote. So diploid condition. Zygote is a diploid condition. Then here no resting stage of zygote. No zygospore stage. This zygote immediately undergoes to mitotic division. Then forms the embryo. Then embryo develops into sporophyte. So diploid condition. In sporophyte, spore mother cells are present. In sporophyte, spore mother cells are present. This also 2n condition. Then spore mother cells undergoes to meiotic division. That means reductional division. So results of meiotic division here form the spores. That means haploid spores. These haploid spores may be directly germinate to form a new bryophytic plant body, or in Bryopsida plants it is germinate to form a protonema vegetative stage. Then protonema to secondary protonema, leafy axis. So then adult bryophytic plants. Haploid condition and then diploid 2n condition. Haploid and diploid, gametophytic stage, then sporophytic stage. So these are morphologically distinct multicellular condition. So heteromorphic alternations of a generation, haplodiploidic life cycle. In Bryopsida polytrichum, so polytrichum useful to remove the stones in a kidneys. Polytrichum is useful to remove the stones in a kidneys.